Hello, we are the Group 2. Brace yourselves as we are going to present our final performance task in histopathology laboratory, which includes infiltration, embedding, fishing out and floating out of tissue specimens, H&D staining, and mounting. So, what are we waiting for? Let's start!
Hello, I am Nika Ocahilas and I am assigned for the infiltration process. When the final silene is replaced with molten wax, it infiltrates the tissue, which is also known as infiltration. The most frequent infiltration and embedding media is the paraffin wax. Every tissue type and application may be accommodated by a wide range of infiltration mediums. Factors such as wax hardness, stickiness, and brittleness should be addressed during microtomy. Also, the method and the tissue used. As a general rule, the greater the melting point of paraffin, the harder the paraffin and more difficult ribbonning becomes. 3 wax immersions are usually required to guarantee that no cleaning agent remains in the tissue. The tissue cassettes are transported to an embedding station after the final infiltration. The lower the melting point, the softer, stickier the paraffin, and the easier it is to ribbon during microtomy. Before we proceed, let's check first our PPE. Head cap, glasses, mask, lab gown, and gloves, because remember, it's better to be safe than sorry. Now, let us proceed to the materials. These are the materials we need for the infiltration process. First is paraffin wax. Check. Second is wax holder. Check. Third is forceps. Check. Laboratory thong. Check. And lastly, paper towel. Check. And now we are ready to proceed.
Tissues must be immersed in melted paraffin wax or melted candle for 15 minutes in at least 4 changes in order to ensure complete removal of the clearing agent from the tissue specimen. Step 1. Label each wax holder in a proper order. Heat the candle wax in an oven set at 56 to 60 degrees Celsius until it turns liquid. Make sure to keep the wax warm and keep it from solidifying. Step 2. After heating, transfer the tissues to the first infiltration bath. Forceps must be warmed and place it back into the oven and leave the tissues for 15 minutes. Step 3. After 15 minutes, transfer the tissues to the second infiltration bath. Forceps must be warmed and place it back into the oven and leave it for another 15 minutes. Step 4. After 15 minutes, transfer the tissues to the third infiltration bath. Forceps must be warmed, place it back into the oven, and leave it for another 15 minutes. Step 5. After 15 minutes, transfer the tissues to the last infiltration bath. Forceps must be warmed. Place it back into the oven and leave it for the last 15 minutes. Step 6. Remove the tissue samples from the wax. Place them on paper towels and prepare the tissues for embedding.
The technique of embedding involves enclosing tissues or specimens in a mass of embedding materials using a mold. The tissue blocks require a supporting media in which to be implanted because they are so thin in thickness, and the embedding media is the name for the supporting medium.
The materials that we will be using in the embedding procedure are 1. Paraffin wax, and we can use melted candle wax as a substitute. 2. Tissue cassette or molding box, and we can use matchbox as a substitute. 3. Forceps for the orientation of the tissue specimen. Step 1. After the infiltration process, prepare your embedding molds or matchbox and see to it that it is 1 mold per tissue. Check your paraffin wax or melted candle to make sure it is enough for every tissue specimen. Step 2. Attend to 1 tissue sample at a time. Step 3. Fill the molding box with a small amount of paraffin wax or melted candle for support. Leave for a while to slightly solidify. Step 4. When it is ready, place the tissue samples. Observe proper orientation of the tissues using the forceps. Step 5. Fill the box with the embedding medium until it covers the tissues. Step 6. Leave the box in a cold plate for 3 hours to completely solidify. Step 7. When solidified, remove the embedded tissues from the molding box. And lastly, step 8, label the block.
Flotation and fishing out on a flotation bath or water bath is the intermediate step between cutting paraffin sections and placing them on slides. Simply sticking paraffin ribbons on slides will not work. Thus, a warm water bath allows tissues to relax, smooth out, and remove wrinkles and folds prior to being mounted on a glass slide. The warmth also makes the paraffin stick to the glass slides. Water baths are filled with distilled water, heated to a temperature 5 to 9 degrees Celsius below the melting point of the paraffin. And the water bath is usually kept at 40 to 50 degrees Celsius. This is an optimal temperature range for various types of paraffins. Hard paraffin will require a higher temperature water to relax, while softer paraffins will benefit from a lower water temperature since they may disintegrate at higher temperature.
The materials used in floating and fishing out of tissue sections are flotation bath, distilled water, microscope slides, tissue ribbons, forceps, and slide drying bench. Step 1. Once tissue ribbons are formed, flatten it out at the water bath containing distilled water to allow any wrinkles to disappear. Let it float on the water surface with the trailing end of the ribbon making contact with the water first. Step 2. The water in the flotation bath is set at 42 degrees Celsius or about 4 to 5 degrees Celsius below the melting point of the wax. Take note that a water bath that is too cold will not remove wrinkles in tissue sections, whereas a water bath that is too hot may melt the paraffin and change the tissue morphology. Step 3. Leave the section on the water surface just long enough for it to flatten. Overexpansion can spoil the morphology in susceptible sections. Step number 4. With the use of forceps, split or separate the ribbons into individual or group of sections. Step 5. In fishing out tissue ribbons, immerse the slide vertically near the target tissue section to 3/4 of its length, and pull it out gently to avoid wrinkles and getting any air bubbles underneath the sections. Step 6. Slides are then labeled with the appropriate information. Step number 7. Once finished, place the tissue slides in the oven at 60 degrees Celsius or 5 to 10 degrees Celsius above the melting point of the wax for about 30 minutes.
Hematoxylin and eosin staining is the staining method of choice for histopathology laboratory. This method makes use of two dyes, as the name suggests, hematoxylin and eosin. Hematoxylin stains the acidic structures of the cells, such as nuclei, as color blue. On the other hand, eosin, as an acidic dye, stains the basic structures of the cells, such as the cytoplasm, as color red.
The following are the materials to be used in hematoxylin and eosin staining. Before the actual staining, we must first allow the slide to cool for at least 5 minutes from the slide warmer. After 5 minutes, we will now proceed with the staining by immersing the slide in the following order of reagents. Step 1 is the paraffinization. Clear the paraffin-embedded sections in xylene for 3 changes with 2 minutes each interval. Step 2 is hydration. Xylene is immiscible with stains. Therefore, it is removed with degraded alcohol from higher concentration to lower concentration. We will now immerse it in the bath of absolute ethyl alcohol or alcohol formol for 10 dips. Step 3.
Transfer it to a bath of 95% of ethyl alcohol with at least 3 changes, with 10 dips each. Step 4. After immersing the slide in descending concentrations of alcohol, wash the slides in soapy water and remove excess albumin or impurities using a cotton swab. Then rinse the slide well with a clean water and drain excess water on paper towels. Step 5 is staining. We will now stain the slide in hematoxylin for 5 to 10 minutes. Drain the excess stain, then rinse with water. Step 6. Decolorize sections in acid alcohol for 2 quick dips, then rinse the slides immediately in water. Step 7 is bluing. Immerse the slides in ammonia water until tissue turns blue for 5 minutes. Rinse again in water, then drain the excess water on paper towels. Step 8. Stain the slide with Eosin Y for 1.5 to 3 minutes. Drain the excess stain, then rinse well with water. Step 9 is dehydration. Immerse the slide in ascending concentrations of alcohol. Dip in 95% alcohol for 3 changes with 10 dips each. Lastly is clearing. Dip the slides in xylene for 3 changes and 10 dips each. Then clean and wipe the edges and the back of the slide. This is the end of the H&E process. After staining, we will now proceed to mounting.
In histopathology laboratory, mounting is the last procedure in the series. Mounting is done to preserve, physically protect, and support the stained section for microscopy. In this step, a specimen is mounted on a clean glass slide, then covered with a cover slip. Afterwards, a mounting medium is used to adhere the cover slip to the slide. A mounting media should ideally have a refractive index close to that of the tissue specimen, which approximately is 1.1518. It is important for the slide and the cover slip to be free of optical distortions to avoid viewing artifacts. There are 2 types of mounting media, the resinous and the aqueous media. So now, Val Ayuban will now proceed to the demonstration of mounting.
The materials used in mounting are mountant, glass slides, and tissue section. Once staining is complete, the tissue samples need to be mounted under a cover slip. This is done when histological section needs to be examined for any length of time or to be stored. For this demonstration, we will be using the cover slip method. Step 1. Place a drop of mountant on the cover slip. Step 2. Invert the slide over the cover slip and lower it so that it adheres to the cover slip. And quickly turn the slide over. Then, lay it on a flat surface to allow the mountant to spread. Take note, never press or push the slide at all, because it can damage the section. Step 3. After the mountant has spread to the edge of the cover slip, tap off excess mountant and xylene and wipe the back portion of the slide for neatness. If proper care has been taken, there should be no air bubbles. If many are present, slide should be returned to xylene to remove the cover slip. Thus, it will slip off and remounting is done. Remember, no attempt should be made to pull the cover slip. The slight warming of the slide from below is sufficient enough to make the small air bubbles escape from the slide of the cover slip. At this point, the cover slip should be in the center of the slide with a neatly written label on one side.